Good morning, everyone, and thanks to the organizer and to all of you for a fantastic meeting. I've learned a lot, and I look forward to learning even more in the next few talks. I will be talking about a very recent evolutionary phenomenon, namely the extreme niche construction where humans invade the reproductive niche with their technology. Assisted reproduction has grown very rapidly. There are now over 5 million children alive who were generated by assisted reproduction. But before we get to the novel technology, I'd like to make a couple of points about the hallmarks of mammalian reproduction. Mammalian reproduction involves internal fertilization, drawn here by Leonardo with a typical male-biased angle. We all know that most of the work is actually done by the female. The investment is very asymmetrical because there is placentation, gestation, and even more importantly, there is lactation, which makes the gestation pale in terms of investment, especially in species like the great apes and humans with up to 1,000 days of lactation. But before you can have a pregnancy, you need to find a mate. How do you find a mate? There are very popular paleo fantasies of finding you in a bonobo. But if we ask about human mating systems, one striking finding is that it's extremely variable. You find almost every possible arrangement. In the last century, people were able to survey traditional societies and ask about prevailing mating systems. And the answer was that most societies, most traditional societies, seem to be mildly polygynous, with the pattern, the most common, would be monogamous pair bonding, at least serially so. And this could indicate a long history of a leveling of reproductive opportunities, where you actually share access and you don't let anybody dominate reproduction. Assisted reproduction is a third-party involvement in reproduction, but that too has an old history. In a very interesting study of the mating patterns, the marriage practices in hunter-gatherers, it became clear that arranged marriages have a long history and predate Judeo-Christian traditions or the Neolithic, very likely. And this is evidence for a type of third-party involvement where other people contribute to who you will reproduce and whether you reproduce with someone. Of course, mammalian reproduction involves internal fertilization, and that's mediated by this very strange cell that behaves almost like the transmissible cancers we heard about in the dogs and the Tasmanian tigers. And it's the mammalian sperm, which is the only cell that evolved to travel into another body. It contains one haploid copy of your chromosome, which is totally inactive at the time of transit. It's packed around chromatin. It cannot be expressed. Whatever expression there was happened in the testes of the father who produced the sperm. And of course, this cell faces incredible hurdles based on male-female conflicts. The male germ cell must navigate to the egg. It has to retain its functional potential of the acrosome, which can only burst very shortly before the lucky sperm meets the egg. And it also has to retain the integrity of its haploid genome in the face of all kinds of hostile, be it pH, be it reactive oxygen species, and so forth. The female, on the other side, must protect against incompatible sperm and some very nasty things that might be riding on the sperm, like gonococci or HIV, known to bind to sperm surface molecules and ride into the next host. The female also has to guard against fertilization by the wrong species. Mammal males stand out in general by not being very discriminatory about what they mate with. So that's a problem. You don't want to waste your egg on a useless sperm that will induce a hybrid infertility. And you want to make sure these gametes are not full of pathogens. With regard to the population, you might have an interest in going after sperm from a high-fitness male. You want to make sure that the sperm comes from a male with the correct diploid alleles and that it carries potentially compatible alleles, which will play a big role during placentation. And finally, you want to make sure that the gamete is still functional, that it can still fulfill the dance and the handshake that come in during fertilization. So during reproduction, these sperm have quite a journey. If we take an average-sized large primate and ask ourselves about the length of this female tract, where that really gives the female opportunities for both having sperm competition within her physiology, but also cryptic female choice, the phenomenon by which females might bias the chance of a given gamete of fertilizing her egg or eggs. The average size is about 30 centimeters, so if you think back to the time when half of you was a sperm, it's about a 6K run. And it can happen in a matter of a couple of minutes because it's assisted by female transport. What's striking is this is not a swimming pool with the male gametes having a little sperm competition and whoever swims faster gets there first. It's actually an auditioning, a sequential auditioning of the sperm, and they get taken out by the millions. In a species such as us, about 200 million sperm are inseminated. For comparison, chimpanzees inseminate with 3 billion sperm. So there's big differences in how many sperm are introduced in the vagina or the uterus, depending on the mammals. But what mammals share is a leukocytic reaction after mating and only in the presence of sperm, so vasectomized males do not trigger that. But if sperm are detected by the female, there is a leukocytic reaction, a white cell reaction, and the female floods her uterus with white blood cells that take out sperm by the million. There is million-fold selection, and it's only a couple of hundreds to thousands sperm that make it past the uterotubal junction into the lower oviduct, where they undergo very interesting interactions with the female and a change in their outfit, which we shall address in a moment. And it's only one sperm that meets the egg. So the very famous image that you probably show to all your students, totally wrong. That's in a petri dish in a fertility clinic. It was enzymatically removed all the vestment of the egg, and then you put a couple of million sperm on the egg. In vivo, what you have is more like the Japanese flag. It's one sperm, one egg. How does the female screen? I mentioned the lower part of the fallopian tube, which has a region known as the isthmus because it's very narrow. It has very complex morphology, complex secretions that differ completely from what happens in the uterus. It's more like the nirvana of sperm. In the uterus, the sperm get killed. In the lower fallopian tube, it's a very cozy environment. The epithelium there looks fantastic. There are ciliated cells and more flattish cells without cilia. And mammalian sperm interact with these epithelial surface of the female for hours. They get held. This is an example of a single sperm head being checked out by the females. Hours of very intimate scrutiny. Are there any choice phenomena happen there? We do not know. We just do know that females check their sperm quite well before letting them go one at a time towards the egg. So how do sperm manage this incredible feat of surviving? There's very few sperm that survive the hostile environment of both the vagina, the cervical mucin, and the uterus to get to nirvana in the fallopian tube. Well, you just heard from Ajit Varki that all living cells are covered with a sugar coat, a glycocalyx, and sperm are no exception. In fact, they're extreme. They have a very thick, 60 nanometer thick glycocalyx, and I'd like to suggest this is nanoecology. If you make yourself 1 billion times smaller than you are now, you would see a sugar that's about your size. And these sugars can make chains, and these chains can be branched, and the branches are attached to trees or things that look more like kelp forest and completely cover the membrane. They're only found on the outside. There's an asymmetry. They're on the outside of the membrane, and they mediate all kinds of interaction between cells. In sperm, they seem to mostly mediate sperm survival. There is no MHC molecules on sperm because that would flag it for destruction by female immunity. Rather, they cover themselves with things like defensins that are highly silylated with these self-patterns that tell the females, I'm okay. They are invisibility cloaks, allowing the sperm to sneak by female immune defenses and likely make it to the fallopian tubes. If you remove these last sugars of this sugar coat, they get stuck in the cervical mucus. They don't even make it to the uterus, not to mention the fallopian tubes. So these glycoconjugates, which are combinations of lipids and sugars and proteins and sugars, some of them are made in the testes. But interestingly, most of them are made by somatic paternal cells in the epididymis, where sperm spend days traveling through, maturing, and getting coated with magical invisibility cloaks. And they have to because the female uterus is very well guarded, and takes out all these sperm. It turns out these different immune cell types that you find in different areas of the female genital tract carry specific receptors, and Ajit just mentioned these sialic acid binding lectins, Ig superfamily lectins called Siglecs, that are expressed on many of these leukocytes and are fingering sperm for the presence of molecules that look, okay, you look like you could be me. So at many levels, ranging from sperm development in the testes, where glycans on the sperm are very important for development, to maturation in the epididymis, where they get further coated with a thicker glycocalyx, and these are paternal somatic products. These are needed to make it through the cervical mucins that actually block access to the uterus when there is no ovulation. The uterus is nicely safe, no sperm get there. They only have to get there when there is an egg to fertilize. They then mediate survival in the face of female immunity. They are involved in a single file introgression of these lucky few sperm through the uterotubal junction. They then mediate attachment to the epithelia of the isthmus. And during capacitation, which is a mammal-specific phenomenon, where sperm undergo a membrane change, they ditch the tuxedo and put on a functional, very sexy pajama. They then approach the egg and biochemically etch their way through the hyaluronic vestment around the egg, and only then interact with the zona pellucida, which is, of course, also a glycoprotein. They get through the zona pellucida, and then they fuse with the membrane. So if you do all that, you get a pregnancy. But once you have a pregnancy, we all heard about preeclampsia, and that preeclampsia is really a failure of the fetal cells to remodel the spiral arteries on the maternal side so that mother can exsanguinate into this pool from which the fetal villi take their nourishment. Interestingly, absence of contact with paternal sperm is a risk factor for preeclampsia. So one role of the sperm surface and semen components might be a kind of immune priming of the female. Hey, there is somebody else here that I shouldn't discriminate against too much. So assisted reproduction technologies are younger than half a century. The oldest child is now in her 30s. It's a completely novel phenomenon, and it started out by classical intrauterine insemination or in vitro fertilization, which use millions of sperm. Much more recently, since 1992, we have the possibility of doing ICSI, intra-cytoplasmic sperm injection, which uses exactly one sperm chosen not by the female, but by a technician under a microscope. Usually the tail is yanked off, and the sperm nucleus is directly injected into the egg. These technologies have realized the hope for parenthood for a lot of naturally infertile couples. There's a range of advanced techniques ranging from freezing to culture and so forth. They've also made it possible for same-sex parents to realize the dream of parenthood. So there's been a massive change and a great opportunity for many people who wish to have kids and naturally would not have them. They also allow pre-implantation diagnosis of fetal cells for mutations known to be super dangerous and leading to a lot of suffering and unnecessary suffering. More recently, you've all heard about the three-parent phenomenon and the possibility of genome editing using CRISPR-Cas. These are very interesting phenomena that haven't happened yet. The three-parent thing is happening, where you take fresh mitochondria, you have three parents. It's a germline manipulation that has become a reality. So it turns out that in vitro fertilization and assisted reproductions is a billion-dollar industry as well. And the reagents used are proprietary and usually don't list their contents. It's a large-scope operation. The success rate is surprisingly low, and we do not really know what are in these reagents. A recent study by a group in Denmark found that in addition to the human serum albumin, there were over 100 proteins present in these conditioned media. And my own lab has started looking at this xenosialic acid you heard about from Ajit, and it's present in vitrification solution in which we freeze embryos. It's also present in the solution in which we freeze sperm and in the enzymes used to treat the egg. People interested in the effect of in vitro culture of mammalian embryos, this is Paolo Rinaudo at UCSF, has compared gene expression in embryos generated in vivo, in vitro, or fertilized in vivo and then cultured in vitro, and showed very big differences in gene expression in these different conditions. He's also measured what happened to the growth of the embryo and the placenta, and they differ from normal in vivo conditions. Very recently, his group, this is Paolo Rinaudo at UCSF, has found a sexually dimorphic effect of IVF, just doing exactly what he does as a fertility doctor with human embryos, with mouse embryos, where the females, very late in life, get very heavy and have a dysregulated lipid metabolism. So one concerning idea is that we might be doing things to these embryos in the 5 days that we culture them, like changing their imprinting. And that's very troublesome because the experiment is ongoing. So in summary, I think I've convinced you that female filters, uterine and fallopian, exist, but they're very poorly understood. Art is kind of the logical extension of human niche construction and our tendency to level reproductive opportunities. Art bypasses female choice. Art exposes gametes and embryos to biocontaminants. There's evidence from animal models that this might be a concern. Art is known to be associated with risk, perinatal and postnatal. It's fulfilling a lot of people's hopes, but it's a large-scale experiment that is still ongoing. So with this, thank you very much, and I'd like to thank Feng Ma and Ilan Tekle.